Hello students. So I posted two links here in chat box, one for presentations and one for the recorded sessions. So you can get all the materials in this link and from this YouTube and this Google Drive. So those who are interested, they can access the presentation from here. Right now there is no access to the presentation. After meeting, I'll allow everyone to get the access of that particular presentation or particular video. So let's start. Does anyone have any question from the last two sessions? They can start asking. Or if you don't have any questions, then we'll start today's session. So let me present the presentation. Yes, Arun, you can ask your question in chat box. Please ask. Doubt about it. Yes, Arun. You can unmute your mic also if you have some other doubts. Okay, so let me start. Okay, so this kind of queries you can ask to the NPTEL directly to the NPTEL managers, and you can ask to the NPTEL specific course coordinator. So you can mail directly to them. This type of query is not will be available here. You can mail that this particular specific doubt to the specific course coordinator. So maybe he will help you out for this. So let me present my screen. My screen is visible to all. Is my screen is visible? Just anyone can confirm.
Yeah, it's visible.
So let's start. So for today we'll discuss about nanomaterials for cancer diagnosis and its therapy. So first we'll discuss about cancer. What is cancer? So welcome to the presentation on nanomaterials for cancer diagnosis and therapy. So cancer is a global health challenge and innovative approaches are needed for early diagnosis and effective therapy. So cancer is a group of diseases characterized by the uncontrolled growth and spread of abnormal cells in the body. It can affect any tissue, any organ, or and maybe result in formation of a tumor. These growths are considered either benign or malignant. So each type of cancer is unique with its own causes, symptoms, and method of treatments. So not all tumors are cancerous. Benign tumors do not spread to other part of the body. So causes of cancer. So Any agent that causes cancer is called carcinogen and is called as carcinogenic material or carcinogenic agent. So there are different factors which are responsible for cancer. So the first is genetic factors, which are inherited gene mutations can increase the risk of cancer. Then lifestyle choices like tobacco use, unhealthy diet, lack of physical activity, and excessive alcohol consumption. Then some are the environmental factors also, which are exposure to carcinogens such as radiation and certain chemicals, mostly UV radiations. So, cancer development. So, there are three phases, there are three stages of cancer development. So, the first is initiation. As you can see in the figure also, there is a normal cell only. Then there is a promotion in the cells. Then there is a progression in the growth of the cells in particular things. So, the first one is initiation, in which genetic mutations or changes in cellular DNA occur. Then promotion, and promotion uncontrolled cell growth, and then progression, and there is an invasion into nearby tissues and potential metastasis. That means spread to the distant organs, nearby organs. It can affect nearby organs also. So types of cancer. So there are over 100 different known cancers that affect humans, but most common ones are breast cancer, brain cancer, lung cancer, colon cancer, and prostate cancer. So stages of cancer. There are four stages of cancer here. The stage one, as you can see in this particular diagram, there is cell with genetic mutation. Mutation is certain changes in the gene. So if in case there is a gene and it occurs certain changes, so that leads to the mutation of a particular cell or a particular gene. So then hyperplasia occur, then after some time dysplasia, then in situ cancer, then invasive cancer. So we are talking in the stage forms. Stage 1 cancers are localized to one part of the body, which are usually curable. Stage 1 is curable. We can cure if it diagnosis in the early stages. Then stage 2 and stage 3 are cancers which are locally advanced, dysplasia and in situ. Then in stage 4, cancers have often metastatized or spread to the organs, other organs, or throughout the body. So here is one example for the breast cancer. So in stage 0, the abnormal cells are present but have not spread to the other tissues. Then leads to the early stage. The early stage cancer has spread to the tissues in small area. Then in stage 2 localized, tumor is in between 20 to 50 mm and some lymph nodes are involved or a tumor larger than 50 mm with no lymph nodes involved. Then in stage 3, this is a regional spread. The tumor is larger than 50 mm with more lymph nodes involved across a wider region. In some cases, there is no tumor present at all. Cancer may have spread to skin or chest wall. Then in the last stage, stage 4, distant spread. Cancer has spread beyond the breast or other parts of the body. So we'll look into it detail in some presentations also. So now the nanotechnology in detection and diagnosis of the cancer. This is the main portion which we are going through. So nanomaterials offer promising solutions due to their unique properties and versatility. The nanotechnology can be used for better cancer diagnosis. One of main usage fields of optical nanoparticles is to allow better cancer detection. Then classical methods that are used in iron diagnosis have limitations such as X-ray, tomography, or I think someone is unmuted. I think someone is unmuted. Nasreen Najib, can you unmute your mic? Mute your mic, please. Nasreen. Classical methods that are used iron diagnosis have limitations such as X-ray, tomography, or mammography, require using mutagenic agents on cells that cause cancer too. So to eliminate these concerns, optical nanoparticles and diagnosis is possible technique that can be used. So this techniques works with some special with tumor cells and optical nanoparticles can be detected. So here's one comparison of traditional and nano-based methods of cancer treatment. So as you can see, this is the traditional cancer treatment. It is a nano-enabled anti-cancer treatment. So in traditional treatment, there is less surface area, and poor targeting capabilities, and poor bioavailabilities as compared to the nano-enabled anti-cancer treatment. So in, nano-enabled, higher surface area is there, better targeting capabilities are there, better bioavailabilities are there. So in traditional anti-cancer treatment, this partial killing of cancer cells. They are not 100% eliminating of the cancer cells. But in nano-enabled things, they are profound killing of cancer cells. That can be 80, 90, or even 95% killing of the cancer cells. So moving forward, so these are the applications. So as our topic was diagnosis and therapy of cancer. So, in nanotechnology, there are three things which are used for the diagnosis of cancer. That is quantum dots, nanoshells, and gold nanoparticles. These three things are used to diagnosis of the cancer, and the rest are used for the therapy of cancer. Liposomes, carbon nanotubes, polymeric micelles, dendrimers, and quantum dots. So, we'll look into in details for these things. So, the diagnosis. So, first we'll go through quantum dots. Quantum dots is used in both, like in diagnosis, also in therapy also. So near infrared quantum dots, the lack of ability to penetrate objects limits the use of visible spectral imaging. Quantum dots that emit fluorescence in the IR spectrum, that is 700 to 1000 nanometers, have been designed to overcome this problem, making them more suitable for imaging colorectal cancer, liver cancer, pancreatic cancer, and lymphoma. The next is nanoshells. So another commonly used nanotechnology application is the use of nanoshells. So nanoshells are dielectric cores between 100 to 300 nanometers in size, usually made of silicon and coated with a thin metal shell, usually gold. So these nanoshells work by converting plasma-mediated electrical energy into energy and can be flexibly tuned optically through UV infrared emission or absorption arrays. And nanoshells are desirable because their imaging is devoid of the heavy metal toxicity, even though their uses are limited by their larger size. The next is colloidal gold nanoparticles. The gold nanoparticle is a good contrast agent because of its small size, good biocompatibility, and high atomic number. Research shows that gold nanoparticles work by both active and passive way to target cells, as you can see in this figure. The principle of passive targeting is governed by a gathering of gold nanoparticles to enhance imaging because of the permeability tension effect in tumor tissues. On the other hand, the active targeting is mediated by the coupling of gold nanoparticles with tumor-specific targeted drugs, such as EGFR monoclonal antibodies, to achieve gold nanoparticle active targeting of the tumor cells. So the down thing is the active targeting, and on the upper thing is passive targeting things. So as you can see, in the active one, the cancerous cell and the healthy cells are different. And in passive cancer cells are done by osmotic tension effect. So here's one example, the gold nanoparticle things. So here various shapes of gold nanoparticles are there, which are loaded in the tissue containing cancer cells. Then we use imaging to identify the cancerous cell by using gold enabled markers. So here are some applications of the gold nanoparticles, which are of different types like spherical, rod-shaped shells, labeled spheres. The imaging applications known as X-ray imaging, fluorescence imaging, surface enhanced Raman spectroscopy, photoacoustic imaging, and optical imaging. So their advantages are they are have high payload delivery, generates a strong electromagnetic field, requires strong absorption in the NIR window. In optical imaging, the light used is in the NIR window also. I think someone has raised some query. Your voice isn't clearly audible. Okay, let me reshare my screen. Wait for a while. I'm resharing my screen. Is it visible now? So, is it now visible? Okay. So there's one figure by which is wait, wait, slide 9. Okay. So we'll start again from the slide 9. So is it visible now? Okay. We'll start from slide 9 again. So in slide 9, so okay. So in slide 9, there is a comparison of traditional and nano-based methods of cancer treatments as well. So there are two types of treatments for there, traditional and nano-enabled. So in traditional anti-cancer treatment, there is partial killing of cancer cells as it has less surface area, poor targeting capabilities, and its poor bioavailabilities as compared to the nano-enabled anti-cancer treatment. So in nano-enabled things, the profound killing of cancer cells were there due to its highest surface area, better targeting capabilities, and its better bioavailability. So now we have some applications of nanotechnology in diagnosis and therapy. So we have quantum dots, nanoshells, and gold nanoparticles for its diagnosis, and liposomes, carbon nanotubes, polymeric micelles, dendrimers, and quantum dots for its therapy. And quantum dots are used in both diagnosis and therapy. So this we can call this theranostics, nanotheranostics things. So now we will go for the diagnosis one by one. So we'll go for quantum dots, NIR quantum dots. So the lack of ability to penetrate objects limits the use of visible spectral imaging. Quantum dots that emits fluorescence in the near infrared spectrum, that is 700 to 1000 nanometers, have been designed to overcome this problem, making them more suitable for imaging colorectal cancer, liver cancer, pancreatic cancer, and lymphoma. Now nanoshells. So another commonly used nanotechnology application is the use of nanoshells. So nanoshells are dielectric cores between 10 to 300 nanometers in size, which are usually made of silicon and coated with a thin metal shell that is gold. These nanoshells work by converting plasma-mediated electrical energy into light energy and can be flexibly tuned optically through UV infrared emissions or absorption areas. So nanoshells are desirable because their imaging is devoid of the heavy metal toxicity, even though their uses are limited by their larger size. So the next one is colloidal gold nanoparticles. So the gold nanoparticle is a good contrast agent because of its small size, its good biocompatibility and high atomic number. So research shows that gold nanoparticles work by both active and passive ways to target cells. The principle of passive targeting is governed by gathering of gold nanoparticles, as you can see in this figure. So in passive targeting, which is the gathering of gold nanoparticles to enhance imaging because of the permeability tension effect in tumor tissues. On the other hand, in the active one, The active targeting is mediated by the coupling of gold nanoparticles with tumor-specific targeted drugs, such as EGFR monoclonal antibodies, to achieve gold nanoparticle active targeting of the tumor cells. So moving forward, so this is a figure which is explaining the gold nanoparticle in the tumor targeting things. So here we have some different shapes of gold nanoparticles, then which are loaded in the tissue containing cancer cells. Then we are going for the imaging. In imaging of the cancer cells, gold enabled markers. So here are some applications of gold nanoparticles in imaging and its therapy in diagnosis. So nanoparticles have different shapes, like rod-shaped shells enable spheres, star-shaped, cluster-shaped shells, primarily spheres. Then the applications of imaging are X-ray imaging, fluorescence imaging, surface enhanced Summon spectroscopy imaging, photoacoustic imaging, optical imaging, which have an advantage of high payload delivery, generates a strong electromagnetic field which requires a strong absorption in a near infrared window. So this is the application of cancer diagnosis of some nanoparticles. So the different types of nanoparticles are carbon-based nanoparticles, ceramic nanoparticles, metallic nanoparticles, and polymeric nanoparticles. And this application is in cancer detection and diagnosis, cancer imaging, and detection of cancerous cells. With this property and some example of its cancerous diagnosis. So the next one is liposomes. So as you can see the structure of liposomes, which have hydrophilic core for water and hydrophilic drug, then lipid layer for lipophilic drug and a hydrophilic head. And its application in vaccine therapy, photodynamic therapy, drug delivery and diagnosis. So the liposomes are one of the most studied nanomaterials, which are nanoscale spheres composed of natural or synthesized phospholipid bilayer membrane and water phase nuclei. So because of the amphiphility of phospholipids, liposomes form spontaneously, allowing hydrophilic drug to preferentially stay in the monolayer liposome, while hydrophobic ones form before the multilayer of liposomes. So under the influence of EPR effect, the vesicle of size around 4000 kilodalton or 500 nanometer can be allowed into the tumor by the caps in vessels of the tumor cells. In tumors, they can fuse with cells and internalize by endocytosis and release drugs in the intercellular spaces. And the liposomes up to 100 nanometers easily penetrate the tumor and stay longer, while the half-life of the bigger liposome is shorter because they are easily recognized and cleared by the mononuclear phagocyte system. The liposomes bound antibodies target tumor-specific antigens to ensure active targeting and then transport drug to the tumor cells. With a lot of pharmacokinetic benefits, some liposomal drugs are approved for its clinical therapy. So here one example is there, liposomal form of adramycin, which have been used for the management of metastatic ovarian cancer, where they have shown its appreciable clinical benefits. So here we have some example of the drug which are used in liposome to treat different types of cancers. Like we have drugs like doxorubicin, paclitaxel, urusolic acid, mitomycin C, vincristine sulfate, 5-fluorouracil, citrabin, amphotericin, and different and many more. So diseases, cancers like ovarian cancer, lung cancer, solid tumors, and different type of cancers. So here are some findings by this particular drug or particular disorder. There are some findings. You can go through one by one. So the next nanotechnology used in cancer therapy is carbon nanotubes. Carbon nanotubes are made of single wall carbon. They're excellent materials and have useful applications due to their electrical and thermal conductivity, durability, and lightweight properties. So, as you can see in this figure, so the carbon nanotube is conjugated with anti-cancer drug and its functionalization, which further goes for the drug delivery in cancer tissue or in cancer tissue imaging. This carbon nanotube can also use in diagnosis and in therapy also. So this thing of will also called as nanotherapeutic. So, because of the physical and chemical properties of carbon nanotubes, that include surface area, mechanical strength, metal properties, electrical and thermal conductivity, It is candidate well suited for large scale biomedical applications. Carbon nanotubes also possess a property that allows them to absorb light from the near infrared region, causing the nanotubes to heat up by the thermal effects, hence can target tumor cells. So the natural forms of carbon nanotubes promote non-invasive penetration of biofilms and are regarded as a highly competent carriers for the transport of various drugs molecules into living cells. So due to the suitability of carbon nanotubes, drugs such as paclitaxel are assembled with them and administered both in vitro and in vivo for the cancer treatment. So the next thing is polymeric micelles. So polymeric nanoparticles are the inventions that relate to a solid micelle with a particle size ranging from 100 to 1000 nanometers. So polymeric nanoparticles are collectively known as polymer nanoparticles, nanospheres, nanocapsules, or polymer micelles. And they were the first polymers reported for the drug delivery systems. So the polymeric nanoparticles serves as a drug carrier for hydrophobic drugs and are widely used for the drug discovery. So the polymeric micelle constructed from amphiphilic polymers with a hydrophilic and hydrophobic block, which can perform rapid self-assembly because of the hydrophobic interaction in an aqueous solution. So in the polymeric nanoparticles, which can capture the hydrophobic drug because of a covalent bond or the interaction via hydrophobic core. Thus, to carry the hydrophilic charged molecules such as proteins, peptides, nucleic acids, these blocks are switched to allow interactions in the core and neutralize the charge. So recently, the polymeric nanoparticles have been used widely in the nanotechnology based cancer drug design due to their excellent potential benefits for the patient care. So for an example, adderamycin conjugated nanomaterial was used to treat several types of cancers, where it achieved therapeutic effects to a decent degree. So here are some drugs which are used in the polymeric micelles with different cancer and its findings. So the drugs like apirubicin, genoxol, paclitaxel are used in the solid tumors, ovarian and breast cancers. So the next thing is dendrimers. Dendrimers are highly defined artificial macromolecules with a 3D network that have a high number of functional groups in it. My screen is visible now. Is my screen is visible to all? Okay. So the dendrimers have three layers: the molecular core, the inner layer, and the outer layer. See, you can see the blue dots here. This is the molecular core of the dendrimer, and the red one is the inner layer of the dendrimer, and this purple color material are the outer layer of the dendrimer. In addition to this, they have solubility, viscosity, and micellar properties. Due to their structures, they have multifunctional abilities, making them useful for building next generation nano devices for imaging and its diagnosis. So there are usually two ways to synthesize dendrimers, a divergent method and a convergence method. So in divergent method, in which the dendrimer can grow outward from the central nucleus. As you can see in the divergent synthesis, the synthesis starts from the core one, from the central nuclei. Then it grow outward from the central nucleus. And on the other hand, in the convergent synthesis, where the dendrimer grow inward from the edges and end up in the central nucleus, as you can see in this figure. There are two different things of types of synthesis of dendrimer. So the physicochemical and biological properties of the polymer, including the size, charge, multi-ligand groups, lipid bilayer interactions, cytotoxicity, internalization, plasma retention time, biological distribution, and filtration of dendritic macromolecules have made dendrimer potential nanoscale carrier nowadays. So here's one example, not an example, this is the schematic presentation of dendrimer, which act as a diagnosis and therapy of the cancer. So as you can see, the normal dendrimer is here. Then we conjugate this dendrimer with anti-cancerous drug. Then there are two methods: encapsulation and complexation. So in encapsulation, the drug encapsulated in a particular area. See, as you can see, the cancerous drug is in the central, is nearby to the central core, which is encapsulated in the anti-cancer drug. On the other hand, in the complexation, the anti-cancer drug linkage with the dendrimeric surfaces. As you can see here, the outer layer is covered with the conjugated drug and the dendrimeric things. So moving forward, so there are some application of dendrimers in cancer detection and cancer diagnosis. So the complex is called dendrimer generation. This is platin 5 aminolevinic acids dendrimer, then PAMM dendrimers, which are used in the breast cancer things, bladder cancer, and breast cancer also. Cell cycle analysis applications, drug delivery applications, and diagnosis applications. So the next and the last is quantum dots. Quantum dots are the semiconductor particles with a typical diameter of 2 to 10 nanometers. They are so named because, due to their nanoscale size, quantum effects play a significant role in the light emitting properties. So as you can see, the core, the quantum dot is in blue color, the quantum dot shell is in the red color, and there is one protective shell outer the quantum dot shell. So the mechanism of light emission of quantum dot is under external stimulus. Some of the electrons of the dot material absorb sufficient energy to escape their atomic orbit. So this creates a conductance region in which the electrons can move through the material, effectively conducting electricity. As these electrons drop back into the atomic orbit, energy is released in the form of light, the color of which depends on the amount of energy released. So as you can see, the different fluorescent color of the quantum dots. So these are the quantum dots. So this color emitting things is called quantum confinement. So because of the dots small size, the amount of energy released is relatively consistent from electron to electron, yielding emissions of a single color. So the color is entirely dependent upon the size of the dot. The larger dots, like 5 to 6 nanometer, which will emit a lower energy emission and will give red and orange color. So this the last color is like red, yellow, and the blue one. Like it's from smaller to the larger size. And the smaller dots like 2 to 3 nanometer providing higher energy emission like blues and violets color. So this property is sometimes referred as a quantum confinement. So in cancer diagnosis, quantum dots immunostaining is a potential tool for the detection of various tumors biomarkers such as cell protein or other components of a heterogeneous tumor sample. Quantum dots can gather in a specific part of the body and transfer the drugs to those parts. The ability of the quantum dots to concentrate in a single internal organ makes them a potential solution against untargeted drug delivery and possibly avoid the side effects of the chemotherapy. So the last advancement in the surface modification of quantum dots, which combine with biomolecules including peptides and antibodies, in view can be used to target tumors and make possible their potential application in cancer imaging and treatment. So some studies combine quantum dots with prostate-specific antigen to label the cancer, while other use of quantum dots to make biomarkers that speed up the process with some immune markers, having a more stable light intensity than traditional fluorescent immunomarkers. So, hmm, so the quantum dot was the last one, so we are not finishing this presentation. So if someone has any query or any doubt, so they may ask in the chat box. I'm done with the presentation. If anyone has any doubt, they can ask. So, let me start presenting first. So I'm getting some request for the access to edit the presentation. So after the meeting, I'll provide the access to all the students who have requested to me. We'll get all the presentation and the recorded session in that link. So those students who joined late, they can find the presentation link and the recorded session link in the chat box. When you're going to share all the PPTs. So Neha, I've shared one Google Drive link. Scroll up in the chat box, you will find two links. One is Google Drive link and one is YouTube link. So in Google Drive link, you can find all the presentation which are done here in the last 2 weeks. So maybe you do not have access to that presentation. I'll give the access to the viewer only. So after this presentation, I'll give access to all the students who have requested to me. Okay, I'll send the link once again. Okay, okay. So again, I've pasted the link for the YouTube and the Google Drive. You can access all the presentation and the Google Drive uh, recorded session over here. So let's conclude the meeting then. If you do not have any doubts, okay, I'm getting. Thank you guys. So you'll able to access this presentation after 10 minutes of this meeting. Okay. So good luck with your assignments and exams.